Hi everyone, I'm Martin. As Isabel already said, I work at EMBL at the Electron Microscopy core facility, and obviously because we are part of that institution, we drive forward public deposition of all our data that we collect. And I'm somehow the person that is always responsible of depositing the data, and that means I have very good connections to the developers of the archives and repositories, and I'll just give a little overview of what we do and how we do it. In respect of time, I won't talk about metadata, but focus really on like the applications. Good. So is this moving? Yes. So as Matthew just mentioned, we mostly use EMPIAR because we're an electron microscopy core facility, and what EMPIAR does really, really well, and that's the majority of data sets that are there, is single particle Cryo-EM. And I think the reason why that is the case is because the definition of what is the raw data and what is the final outcome of the experiment is very obvious. So the raw data typically nowadays will be single 2D images, and the single 2D images is not a single image, but it's actually composed of image frames. So there can be hundreds of them for a single 2D image. With different electron dose, each of them, and that has implications into the damage and to movement, so you can do some processing. So that's typically the raw data that is deposited. And the significant outcome of the experiment is the structure of a protein or multiple proteins, depending on the conditions and so on and so forth. So that's rather clear. So it's clear what is the raw data, and it's clear what's the final result. And I think this is what makes it easy to handle. I will show you a couple of other examples where already the definition of what is the raw data and what is the final outcome gets tricky, and obviously that then makes it tricky to deposit and archive this data properly. The nice thing for single particle Cryo-EM data is that you can also upload what's called a workflow to EMPIAR, which will basically cover all the parameters and details of the process and how you get to the volume, to the structure of the molecule from your raw data. And again, this is not trivial. It's already not trivial for single particle Cryo-EM, but for other more complicated approaches, it's really, really worse. So I want to talk to you mostly about volume EM, and then volume EM is a broad field. There's several different approaches how you can reach volumetric data, how you can get volumetric data out of your electron microscopes. There's a great community, and we've been working on disseminating all these techniques and also interacting and collaborating a lot with the public archives on that. But you can see already there's like 5 or even more different techniques under the volume EM tag. And each of them has different metadata associated. Each machine works in a different way, and so you immediately realize that there's a huge mess of experimental data and metadata that you somehow need to keep track of. That's when it gets tricky. So my role was that there is a couple of categories when you upload data into EMPIAR. And this is a screenshot. This is what's currently available, which covers many aspects of things. But for me, when I started uploading my first data into EMPIAR, it wasn't clear to me, okay, where does my volume EM experiment actually fit? So that was the first contact point to the developers, and we discussed, okay, what category does make sense, and where would you actually basically assign your data to? Which category do you deposit the data to? So that was kind of the first interaction, and that's also not always trivial because not every type of data fits in these categories that are meant to be generic, but sometimes it just doesn't match, and then that would be the time to get in touch with the developers. What makes it even worse is that your experiment is not only about acquiring the data and acquisition, and if it's multimodal and you have maybe some light microscopy, spatial registrations of your data sets with respect to each other, but actually your experiment starts even earlier with preparing a specimen. And again, together with the volume EM community, there has been a very nice widget that was developed, and it's now available for EMPIAR upload, where you can actually specify your specimen preparation workflow according to the standards that we've seen already, and then you can link that to your publication. And I think this is very, very valuable. So but with every step that you think about you want to deposit in your experiment, it gets more and more convoluted. Okay, so let's get to the discussion. What is raw data? Or I would actually call it relevant experimental data, right? So I have just two examples in tomography. The definition, what is raw data, is rather straightforward in tomography, because you acquire individual 2D images at different tilt angles. If you do that in Cryo-EM, you can use the same approach as for single particle. You can just deposit the frames, because that's really what comes out of your microscope. But then what else is relevant for your experiment? I mean, is it really just the protein structure that you get at the end, or is it the morphology that you want to describe? And for this, is actually the reconstructed volume interesting that you can get from these individual images? Or is it a reconstructed volume that has been processed somehow, maybe denoised using AI techniques or filtered? Because that's actually the volume where you've been doing the picking or the segmentation of relevant morphologies. Or is it the individual subvolumes that you use for further analysis? So that already gets tricky. And I think this is really up to the individual experiments to decide what is the relevant experimental data. I don't think there's a general rule, but maybe if we collect more and more data, we can maybe come up with a bit more general guidelines on what is really relevant for the different types of experiments. And the same is actually true for slicing volume EM, where you actually acquire a lot of 2D images of individual slices, and then you analyze the volume. So is the raw data, which is unaligned slices, or even tiles, if you have multiple tiles in 2D that you acquire, is it really useful to have that available, or is it enough to just upload the aligned volume, because that's what you look at? And then that's where you do your segmentations, because actually the segmentations contain all the relevant context of the different morphologies of the organelles and so on and so forth. So you see, it's even deciding what part of the data is relevant for the upload is not always straightforward. And then obviously, when you look at the different depositions, and I'm not even looking at volume EM depositions, it's not the strategy can be different. So for example, what you see here is there's 3 different depositions for single particle cryo-electron microscopy, and basically each deposition is a different variant of the experiment. So I think in this case it's clear, but then it needs to you need to make sure that these 3 depositions are actually linked and describing the same type of data. Other depositions in the volume EM field, for example, deposit every single volume as a different deposition with a different ID. Or you could think of, okay, because actually it's not really clear what is the actual experimental difference between these volumes. So you could probably also have deposited them all together, and just in the description of your data describe what is the different conditions, or maybe it's just biological replicates and they actually show the same thing. So yeah, the way you upload data also depends on what you want to show, and this is currently done differently. So these are just 2 random examples that I took from, I think, the first 2 pages of EMPIAR depositions. If you go there, you'll find them as well. So what I used to do for the depositions that I did, for example, for tomography, is that I would put the individual tilt series, the raw data, together with the reconstructed volumes into one deposition, because I think that belongs together, and that's how I handle it. But again, there's no strict way of dealing with it, and I think we should just probably wait a little and see what different depositions look like, and then maybe come up with some guidelines how to do that. The last thing I want to touch on is segmentations. And segmentations for volume EM experiments is key, because that's what contains the volumetric information. There is the EMDB segmentation format that internally segmentations are dealt with. And typically segmentations you would visualize like this. So you visualize the volume, slice through it, and then you show some 3D rendering of your segmentations. This is a plankton cell collected in the ocean, and you can look inside. You visualize the organelles and so on and so forth. That's cool. That's what you typically visualize, but that's only, I would say, half the story, because I mean the key information that you have in there is actually what is what. And this is something that a movie like that doesn't really show you, but actually you need to annotate, and it needs to be clear what is what. And this is actually what the segmentations on EMDB and also EMPIAR focus on, that each of these segments is properly annotated, and it's linked with the ontologies. This is what we already heard before, that the object we're looking at is directly associated with what we're looking at. So this was a recent study of this plankton cell, and I got in touch a lot with the people behind the segmentation format, and there was a lot of improvement on that side, because right now actually you can get it both together. So what you see on the next slide is this is a visualization of one of these EMDB segmentation files. So it actually contains exactly what we've seen before. You can visualize it in Chimera X. This is an open source software that's used a lot. It has a very nice 3D rendering, all the fancy stuff where you can put different lighting and everything. So the EMDB segmentation format is now supporting this properly. Properly means that the colors are the same as in the original segmentation. The spatial relation is the same. So you could probably think of having done the segmentation in a downsampled volume, but still it's registered in the correct way now. So this is part of the development that we've done with that particular data. And the most important thing actually is that this segmentation that we're just looking at internally contains all the metadata. Yeah, so it was scanning electron microscopy data set. It's this type of plankton. It's collected in France in the Mediterranean Sea. And then each of these segments is, labeled. Chloroplasts in there, there's chromatin in there. Has eye spots, right? So, and the nice thing is that this is all part of that segmentation file. So you could think of looking for mitochondria collected in the Mediterranean, and because we deposit it that way in the archive, you will find our data set, because all these annotations are available together with the segmentation. And with this, I'm already coming to my conclusions. So what is EMPIAR used for? And I think this was already mentioned before. So the focus is we want to make the data available and searchable. Okay, so we want to make sure that with whatever approach, whatever annotation you come to the data set, you will be able to find it. So this is what we always need to keep in mind. One thing that I've found out during the several depositions, and I would say exotic depositions, so no cryo single particle EM data, is that during the deposition process, the interacting with these large data sets can be very tricky. So I am obviously not suffering from upload times, because we have a direct fiber line to the archive from our institute. But I would say, from my experience, I would now try to deposit as late as possible, because you have something like a review process, and reviewers will ask you to add more data to an existing data set or maybe modify it. And that is, with the current procedures, is extremely tricky for technical reasons. So if you want to deposit and you don't need to show it to the reviewers before the paper is in its final form, I would suggest it makes your life much easier to do that as late as possible in this process. Fast interactive browsing streaming modes is currently not possible with that type of data. And again, this is for technical reasons. And I think in the next talk covering new generation file formats, you will hear about our approaches on handling this, and maybe this will in the future be integrated. But you really need to make sure when you upload your data to EMPIAR or to one of these archives, then it's really about the archiving. It's really about that the data is accessible. It's not extremely performant, and there's no super nice visualization. Hopefully yet, we might be able to provide you with solutions there in the future, but that's a bit tricky at the moment. But the good thing is, we can reach out to these people that develop on the service side. And typically they are quite open-minded, and if you have a suggestion or ideas, then there's a good chance that these things get implemented. And with this, I want to thank you. So mostly the first lines here is actually EMPIAR developers. We've been in touch a lot because of my exotic contributions and their positions to EMPIAR, and they've made it available that you can access it through there now. Paul was extremely helpful in looking into the segmentation formats, and I think this is an extremely valuable add-on that adds a lot of information to the data. Tom worked on the implementation of this Chimera X viewer, and obviously, without the scientists that create the rawest data, so they come with the sample that we image in the end, this wouldn't be possible. So this is the most exciting part is actually working with the scientists and then generating this data.